Hello everyone, and welcome back to our study of the cardiovascular system. Before we take on the task of understanding cardiovascular physiology, we need to lay down some foundational concepts. We will be looking at some simple fundamentals in regard to hemodynamics, which is the study of fluid dynamics within the cardiovascular system. To begin, let's look at the architecture of the heart. Now, normally the heart functions as two separate and distinct pumps, with blood on one side not mixing with blood on the other. An exception would be in a person who has a patent foramen ovale. Some deoxygenated blood from the right side of the heart may mix in with the oxygenated blood on the left. So let's recall that there are two main circulatory routes. We have the systemic circulation and the pulmonary circulation. The heart is the pump for each, and these two circuits are interconnected in series with the two pumps of the heart. Now let's take a look at a schematic, which I have borrowed from someone else. This is a simple line drawing that clearly shows what I have described. So let's take a look at the schematic and see where it takes us. Starting on the left side of the picture, Dr. Chandas starts us on the right side of the heart, with two great vessels bringing deoxygenated blood into the right atrium. These are the superior and inferior vena cava. From the right atrium, the blood passes through the AV valve into the right ventricle. And then from the right ventricle, it passes through the semilunar valve, exiting the heart through the pulmonary trunk. Pulmonary trunk branches into the left and right pulmonary arteries. And the pulmonary arteries bring the deoxygenated blood to the lungs to drop off carbon dioxide and pick up oxygen. The blood returns back to the heart through the four pulmonary veins and enters the left atrium. The blood moves from the left atrium through the AV valve into the left ventricle and is then pumped out of the left ventricle through the semilunar valve into the aorta. So we can see very clearly from this schematic that the heart is two separate pumps interconnected in series. Focusing on one of these pumps, we can see how blood is directed in one direction and one direction only. As blood enters the atrium, the AV valve is open, allowing blood to flow freely into the left ventricle. We can call this valve the inlet. The atria will contract, pushing the rest of the blood left in the atria into the ventricle. Once the ventricle contracts, the AV valve is forced shut and the semilunar valve is forced open. Blood is ejected through the semilunar valve, and we can call the semilunar valve the outlet. When the ventricles relax, the semilunar valves shut tightly, preventing any backflow of blood into the ventricles. Thank you, Dr. Chandas, for your most helpful diagram. Now you might be saying, okay, I've got this. There's nothing new here. And that is good. It means you have studied and you understand the basic mechanics of the heart. But let's go a little further. We know that the heart contracts, and when it does, it closes down the chamber, decreasing the space within it. Let's focus on the ventricle, because ventricular contraction is the most important part of the cycle. Compare the heart to a rubber pipette bulb. You remember Pasha pipettes from biology lab, right? Well, when you press a pipette bulb together, you are pushing the air out of the bulb, much like how the ventricle pushes blood out when it contracts. But that's not the full use of the pipette. What happens when you let go of the bulb? This is where the power lies. The pipette bulb develops suction, and we can use this suction to draw up fluids. Well, the same thing goes on in the heart, and that's because the circuits are a closed system. This suction is very important, and we will revisit this concept when we look at coronary circulation and venous return. For now, let's look at how a plunger pump system works to pump oil through an engine. And again, I have borrowed someone else's hard work to help you learn. Now notice what happens as the plunger pushes the oil, increasing the pressure within the chamber. See how the ball at the inlet is forced into its groove, closing down the inlet, and that the ball at the outlet is forced up away from the outlet, allowing oil to be ejected from the chamber. When the plunger is released upward, a suction action occurs, and the ball at the inlet is pulled away from its groove, allowing new oil to enter the chamber. And at the same time, the ball at the outlet is sucked into its groove, blocking the outlet and preventing backflow of oil into the chamber. And so we can see the usefulness of the pump mechanism analogy in learning about the heart. It makes you wonder if the inventor of this pump actually studied mechanics of the heart. So the last thing I want to go over in this video is how the microscopic cardiomyocytes work together in unison to accomplish the task of pushing 5 to 6 liters of blood through the body every single minute. Cardiac muscle cells have specialized unique features that help them to accomplish this task. These cells do not function independently from one another, and they are tightly bound together at the intercalated discs through membranous interdigitations and desmosomes that bind the membranes together. The membranes are also joined together electrochemically through gap junctions. These gap junctions allow cells to share the movement of ions and therefore the electrical impulses that are transmitted through the myocardium at incredible rates of speed. And the most striking feature of the myocardium is the fiber arrangement of these cells. Arranged in a circular and spiral pattern, these interconnected fibers pull on each other and squeeze the chambers. This squeezing and twisting that the heart goes through with every beat is similar to the effect of wringing out a towel to get all of the water out. One last thing that I want to mention, and I will leave you with a question. During ventricular contraction, the heart wall is squeezed, and the coronary vessels that supply blood to the heart wall are compressed. How does a fresh supply of blood get to the myocardium to replenish these cells whose work is never done?